today by painting a picture in your minds. Think about how we as humans, or how squirrels or birds or almost any other sort of animal builds a home for themselves in order to protect them from their external environment. Now, I want you to keep that same picture in your head, but make it thousands and thousands of times smaller, and instead of animals living in there, it's inhabited by bacteria. These tiny homes made by and for bacteria are not some imaginary concept. They're what's known as a bacterial biofilm. Like ourselves, bacteria must make a home to protect them from the harsh conditions in which they live. And the formation of these homes, or biofilms, are actually what have allowed bacteria to colonize many of the harshest environments our planet has to offer. One of these environments is particularly important to us, and that is the human body. Believe it or not, 80% of bacterial infections exist within a biofilm state in the human body, as biofilms allow them to survive and persist because it protects them against our immune systems, natural defenses, and even in the presence of deadly antibiotics. Now, you may have guessed it. An infection that can evade all three of these factors might be rather hard to mitigate, and you would in fact be correct. That's why we must research these homes that bacteria make in order to help prevent and break down existing biofilms, to assist with infection control and help get folks back to a happy and healthy state. And that's what I do with my research. I study a specific protein involved in biofilm formation in C. diff, a bacterial species that is the #1 cause of hospital-acquired diarrhea that can also cause more severe infections such as toxic megacolon or pseudomembrane colitis, both of which are life-threatening infections of the gastrointestinal tract. Now, think about how we commonly make a house out of wood. Bacteria employ a similar process, except they use long chains of sugars that have been linked together known as polysaccharides. The same way that we must break down a 2x4 plank of wood to the proper sizes, bacteria must cut up these polysaccharides in order to build themselves a home. And the enzyme that I study actually cuts up these polysaccharide chains so they can be released into the external environment and form a biofilm. I employ various scientific techniques in order to experiment with this enzyme to expand our knowledge of the process of biofilm formation. The goal of this research is to understand how this enzyme is involved in biofilm formation, with the hopes to apply this knowledge to produce new and improved methods to prevent and break down existing biofilms during an infection, with the hopes to help our immune system, natural defenses, and antibiotics do their job and fight off infections. Thank you for listening and have a great day.